Or good afternoon, good morning, good evening, depending on where you are. And welcome to the RNA Collaborative Seminar Series, today hosted by the University of Michigan. My name is Mats Ljungman. I'm the co-director of the Center for RNA Biomedicine, and I'm excited to introduce to you today two up-and-coming assistant professor, Sue Hammoud from Department of Human Genetics, and Justin Colacino from Environmental Health Sciences. Before we get started, let me remind you that any questions you have, please type it in by clicking on the Q&A icon on the bottom of your screen. And with that, I'm going to introduce Sue. So Sue is an assistant professor in human genetics at the medical school. She's a specialist in the germ cell development. She got her bachelor degree at Wayne State University in Detroit, and her PhD degree in molecular biology from the University of Utah. The title of her talk today will be Same, Different. Single-cell RNA-seq identifies conserved and divergent features of mammalian spermatogenesis. Welcome, Sue.
Okay, so my lab is interested in understanding how do you make actually a developmentally competent gamete and the process of spermatogenesis. And just to give you all a brief introduction about sperm production and how it occurs in the testes, of course, spermatogenesis occurs within the testes, as you can see here, and particularly within a structure known as the seminiferous tubule. And this is the spaghetti-like structure within the tubule. And if you take a cross-section of actually a testicular tissue or a seminiferous epithelium, you find this very unique organization of the seminiferous tubule, where you have somatic cells that line the basement membrane, and somatic cells are in the interstitial spaces, whereas the germ cells are actually oriented from the basement membrane and actually move inward as the differentiation process progresses. And so, of course, ultimately, fertility relies on the presence of spermatogonia, which are necessary for maintaining reproductive potential throughout an individual's lifespan. And these cells undergo a series of mitotic and meiotic divisions, and eventually leading to these haploid round spermatids that actually undergo morphological changes to give rise to these elongating spermatids, which continue their differentiation process in the vas deferens of the testes to make these mature-looking sperm that are necessary for development. And what we know is that sperm production is a largely conserved process. So you have through the same series of mitotic, meiotic, and post-morphological changes that occur in sperm. But what we do know is that this process actually varies, the duration of the process actually varies across species, where you have this process takes about 35 days in mouse and 74 days in human. We also know that the organization or the differentiation trajectory of the germ cell population is actually very different across species. And so in the mouse testes, a very, very, very small fraction of the cells are the undifferentiated spermatogonia, which are shown here at the top of the pyramid. And these constitute about 0.3% of the total cells within the testes. And they undergo a large number of transient amplifying divisions, which are known as the progenitor cell populations or differentiating spermatogonia, and then undergo 2 rounds of meiosis and spermiogenesis before they give rise to mature sperm. So in any particular cell in the mouse testes, this needs to undergo 12 divisions, and approximately 1 single cell gives rise to 4,000 mature sperm a day. In humans, this process is actually a little bit different. You have a larger number of spermatogonial stem cell pool to begin with. And so therefore, the transient amplifying divisions that need to occur in human are actually much far fewer than they actually occur in mouse. And as a result, you need to undergo 5 divisions before you give rise to sperm. However, this process in human is a lot more inefficient. And so one particular, what you have is about 4.4 million sperm produced per gram of testes per day. And so what we also know is that this process, there's both intrinsic and extrinsic factors have varied across evolutionary time that are governing this differentiation process. And what really shed light onto this was these cross-species transplantation experiments that were done when they isolated spermatogonial stem cells from the rat testes and transplanted these into the mouse testes. And what was really surprising at the time is that although you're bringing in rat spermatogonia into mouse, you could actually complete spermatogenesis and make mature sperm, and these mature sperm are actually fertile. However, when you bring in primate samples, primate spermatogonia from macaque or human, you transplant it into the testes. These cells colonize the seminiferous tubule in the testes, but they can never actually differentiate to make differentiating spermatogonia, nor can they enter into the process of meiosis, which has suggested that differentiation and meiotic entry has evolved across species. And so we're interested in understanding what are the conserved and diverged features of the spermatogenesis program. But in order to do this, we need to be able to compare across species and to compare across equivalent cell types to better understand this process. And so some of the limitations have been in the field, at least for the primate tissue, is that there's no known cell surface markers that we can use to purify all the different somatic and germ cell populations. In mice, of course, we could bypass some of these problems by using transgenic lines to sort of enrich for these different cell types. And so the way to embark on this, we decided to take an unbiased approach, which is single-cell RNA-seq, to provide from all three different species. We take testes, dissociate them, sort out live cells, and then do drop-seq analysis. And this approach allows us to analyze in an unprecedented approach the gametogenesis program, allows us to identify rare and key developmental transitions, and really not rely on a priori knowledge in order to assemble the differentiation program. And also we could begin to learn intrinsic and extrinsic pathways of the germ cell differentiation program. And just for the beginners in the audience, especially the new incoming graduate students, there's a lot of hype about single-cell RNA-seq analysis and approaches to use the single-cell RNA-seq. In particular, my lab has been interested in applying Drop-seq to analyze thousands of cells. And the technical advance with Drop-seq is this molecularly barcoded oligonucleotide bead that has millions of oligonucleotides that are attached to each microparticle. And within each oligonucleotide, you have three important features that are important for the single-cell RNA-seq process, but also important for parallel processing of thousands of cells at a time. And the first important feature is the cell barcode, which is shown here in the different color codes that is present on each individual bead. And so therefore you could capture all the mRNAs that are attached to a single bead are presumed to be derived from a single cell if your dilution process is working accurately through the flow cell. And what you have then are these gene barcodes, which allow you to count the number of transcripts for each individual gene. And then you have these oligo-DT primers that allow you to capture polyadenylated transcripts. And so just to briefly explain how Drop-seq works is that you have these microparticles that have oligonucleotides attached to them. You flow in cells and you flow in oil and lysis buffer, and you basically form these little oil droplets. And again, for your estimated input is that every cell, 1 in 10 cells actually have a bead and a cell together in a single droplet. And so when the cells are actually in a droplet, they lyse, the RNA attaches through these oligo-DT nucleotides, and then you could actually get reverse transcription to occur on the bead, and then you actually sequence the mRNA, the cDNA samples, sequence the cDNA, and by using the cell barcode information, you could begin to deconvolve the transcriptome of each individual cell. And so to visualize the single cell RNA-seq data, what we typically use, and I'm going to be visualizing a lot here in this talk, are these PCA plots, which are the principal component analysis, which reduces high dimensionality data into 2D space. And what I'm showing here is that a PCA plot of a random cell type, and what is featured here are these individual circles here represent a cell and its transcriptome. So cells that are more closely related tend to occupy similar or close, are usually occupied in very close proximity of each other within the PCA plot. And these are usually used to define distinct cell types. And so the general idea here, again, the most important feature is that similar transcriptomes should cluster together and to define particular cell types. And so when applying this on 3 different testes from 3 different species, from human, macaque, and mouse, we identified initially 4 major germ cell populations that are illustrated here. These are spermatogonia. So these are the cells that undergo mitotic divisions. Spermatocytes are the cells that are actually undergoing meiosis. Round and elongating spermatids are the ones that are undergoing morphological changes or terminal differentiation. And then you have this cluster of cells here, which ends up, I'm shading in gray in all three species. These tend to be the somatic cells, which are quite distinct from the germ cell populations when comparing them initially to germ cells. But if you actually do reclustering of these somatic cells, you could identify that there's a large number of different somatic cell types that are present within the different testes. And also the proportion of these different somatic cell types vary across species. So, however, now we have transcriptomes from at least 14,000 cells, 20,000 cells from macaque, and 30,000 cells from mouse. We could begin to compare in an unbiased manner. First of all, how did the spermatogonia behave in the different germ cell populations, and what is the germ cell developmental trajectory, and how does it vary across species, and also begin to explore conservation within the somatic compartment as well as variations within the somatic compartment. And so really to begin with, what we focused on are looking at some of the global features or molecular attributes of these cell types. And this is not only just to look at, the number of genes that are being detected in each cell, but also to try to see whether we recapitulate previous findings that are known in the field from bulk RNA-seq data or histology data that's been demonstrated in the 3 different species. And so the first thing is we look at number of genes detected per cell. And at the bottom on the x-axis, you'll find the different cell types that are actually identified in each of the different species and the number of genes that are actually detected. And one thing that's important to note is that germ cells have been previously shown, or the testes in particular is known to have a large number of gene diversity or transcript diversity compared to all other tissues, but also the germ cells themselves have a higher transcript diversity than the somatic cells within the testes. And this is sort of recapitulated here in our 3 different species. What we've previously shown in our bulk RNA-seq data from mouse is that in mouse at least, the spermatocytes and spermatids, which are the meiotic and post-meiotic cells, tend to have a larger gene diversity than the spermatogonia and spermatids. And this is also recapitulated here from our single-cell RNA-seq data. This feature is actually conserved in macaques, but is actually not observed in humans, whereas humans, the larger gene diversity is actually observed in spermatogonia, and then the number of genes detected drops during differentiation. And so this recapitulates previous findings that we've observed and also extended the conservation of this feature to macaques, a lack of the observation in human population. The second feature that's pretty classic to spermatogenesis, or in particular meiosis, is meiotic sex chromosome inactivation. So in males, because you have XY chromosomes, those cannot completely pair along. Pairing can't happen along the X and Y chromosomes. So what happens is that these 2 chromosomes are set aside in these pachytene spermatocytes, and they tend to be silenced by the sex body formation during the meiotic process. And so what we know in mice is that the number of these XY chromosomes should be silenced. And we know in mice from our single-cell RNA-seq data, there doesn't appear to be very much X chromosome transcripts that is being detected specifically in the spermatocyte population. The number of X chromosome genes are actually reduced in spermatocytes in both human and macaque, but this reduction or silencing is actually to a smaller extent than what we actually see in mouse. And to us, this was really intriguing because we wanted to know whether human and macaque have incomplete silencing of sex chromosomes, and are there certain genes that actually escape MSCI in the 2 organisms. And so what we did is actually then compare the transcriptome of spermatocytes and spermatogonia. And so what we required is that the genes that escape on the X chromosome, in order to call them escapees, they have to be expressed at a baseline at a certain level, and the level of expression has to be 2 times higher than what is observed actually in the spermatogonial population, suggesting that these genes are newly transcribed in the spermatocyte population. And so in human, we identify about 20 genes that escape MSCI. In monkeys, this is about 14 genes. And in mouse, using similar thresholds, we identify a single gene that escapes this meiotic sex chromosome activation. And so to confirm that there is a possibility that these genes are actually newly transcribed within Pachytene cells, what we did is actually use intrinsic fish or intrinsic designed in situ hybridization and targeted our probes to intronic sequences in order to capture newly transcribed RNA within the cell. And so we validated this in monkeys, and this is what you're showing here are a cross-section of the testes. And these three cells here are Pachytene spermatocytes within the testes. And the reason why I say these are Pachytene spermatocytes is they have the classic gamma H2AX marker, which marks the sex body, which is the X and Y chromosome. And then what we find is that overlapping with the sex body, we actually find these distinct puncta within the cell, suggesting that these are intronic reads that are being derived from the X chromosome. And there's a single puncta under each of the different A's. So confirming that there is new expression happening in these Pachytene spermatocytes. And so what this analysis really tells us is that on a global scale, when you're looking at general features of spermatogenesis, there appears to be differences across just the molecular attributes of the cells and as well as potentially transcriptional regulation of these cells. So in order to get a deeper dive into the transcriptional variations that are occurring across the germ cell differentiation trajectory, we wanted to combine the three individual species that we've analyzed into a single, put them on a single scale in order to directly compare molecularly equivalent populations or homologous populations across species. And so to do this, we first focused our analysis on the spermatogonial stem cell population, which comprises both differentiated and undifferentiated spermatogonia across the three different species. And so what you're seeing here is a clustering or a consensus clustering of the spermatogonia from the three different species. We identify 6 different molecular clusters of spermatogonial populations, and using known markers that are previously identified in the field, we could annotate these 6 different molecular subtypes of spermatogonia. And so using undifferentiated spermatogonial stem cell markers, we could say populations 1 and 2 appear to be undifferentiated spermatogonia, 3, 4, and 5 are differentiating spermatogonia, and population 6 are expressing a lot of the early meiotic genes. And so this happens to be a preleptotene or a cell that's committing entry into meiosis. And so this is really exciting that we get a defined hierarchy of differentiation. And so when we were looking at this, we were interested in understanding what is the contribution of the individual species to the different molecular clusters of spermatogonia. And one thing that was really striking that we noticed is that there's differential species contribution to each of the different molecular clusters, but the differences were actually expected based on our spermatogonial stem cell hierarchy. So in human, we know that there's a larger spermatogonial stem cell pool, smaller number of transient amplifying divisions before you enter into meiosis. In mice, the stem cell pool is actually very small. Transient amplifying divisions are very large. And so what you can see here is that the human and macaque are heavily occupy the first 2 clusters of the spermatogonia trajectory, whereas mouse is heavily occupying clusters 3, 4, and 5, which are the transient amplifying divisions within the mouse testes. And so using this information, now that we've aligned molecularly equivalent cell types across the different species, we could actually come and compare, identify conserved markers that identify populations across the 3 different species. And this is shown here by PWL4. is highly expressed in spermatogonial population 1 in human. It's also a really good marker in macaque and a good marker also in mouse spermatogonial stem cell population. However, there are other markers that we identify for population 1 that only work, for example, in the primate species, but are really not good markers for mouse. So basically, we've identified a large number of conserved and diverged markers for these 6 molecular subtypes. And most importantly is that for the first time in the spermatogonial stem cell field, we could actually reconcile the previously described histological populations of spermatogonia that are illustrated here as a dark pale, and B, with different molecular subtypes. So we're providing a molecular signature for each of these histological cell states, and we could link molecularly equivalent histological states across species. And so this is going to be really important for understanding and providing information on expanding spermatogonial human and primate spermatogonial stem cells in vitro. These efforts have been ongoing for decades. However, these cells actually end up dying because a lot of the differentiation signals or self-renewal signals that are provided are based on what we know from mouse data. And so to understand to what extent is the germ cell differentiation trajectory conserved beyond spermatogonia across the 3 species, we've performed a very similar analysis, but this time we actually aligned the 3 independent from meiotic to post-meiotic germ cell populations individually across the 3 different species, and then bin these different pseudotime, basically generated an independent pseudotime for mouse, monkey, and human, and divided the pseudotime into 200 equivalent bins in each of the individual species. And then we performed pairwise correlations between mouse and human across these different pseudotimes. And so what you would expect to see actually, if the differentiation trajectory is mirrored in both populations, you'd expect to see high heat on the diagonal and high correlation between the species. What we actually notice is that when we compare between human and macaque, or human and mouse, or mouse and macaque, this high heat on the diagonal is really not observed. So there's a lot of maneuvering away from the diagonal when comparing across species, suggesting there is heterochrony exists across the spermatogenesis trajectory across the 3 individual species. And so this makes it really hard to compare molecular states or meiotic or post-meiotic processes across the different germ cell populations. And so in order to do this, we had to align or create a universal pseudotime for these 3 different species. And so what we did here is use the principal curve, created this using a principal curve and aligned the pseudotimes to the 3 individual species, and came up with a universal time that we used afterwards to compare the aligned pseudotimes across species. And now when looking at this correlation heat map between human and macaque, is that you see there's a high correlation across the aligned pseudotimes across species. And from there, now we can begin to explore which gene modules or gene pathways vary across species and across cell types. And this is just a k-means clustering of the genes across the 21 different bins that were identified from the pseudotime. And what you can see is that a large number of genes, when comparing the human and macaque, have these horizontal lines connecting them, suggesting that these are the same genes that are expressed in the same phase at the same time in germ cell development across species. However, you do see in any particular case that there are variations in temporal expression of certain genes across the differentiation trajectory. And this is actually worsened as you increase evolutionary distance comparison across species. And so this is what you're seeing here. You're seeing more lines off the diagonal. So when you're looking at the genes that are actually not connected across species, these tend to be DNA repair genes or genes involved in RNA splicing, as well as RNA binding proteins across species. And so the next thing we wanted to compare are the global correlations across the differentiation trajectory and using the human as a reference. And so what we can find is that the correlations in the spermatogonial stem cell populations tend to be much higher when comparing human spermatogonia to mouse and monkeys. However, the global gene expression correlations using one-to-one orthologs actually decreases during the differentiation trajectory. And consistent with this decrease in correlation, you have an increase in species-specific gene expression that you're actually observing here. And so this tells us there is specialization and differences in the differentiation program that we need to learn in order to understand what are the molecular regulators of these different pathways that are going to be important to turn on and off during in vitro gametogenesis for each of the different species. And so just to speed up a little bit, sorry for time, and we did the final comparison was to look at the somatic compartment across the 3 different organisms. And we know histologically that the vast majority of somatic cells are actually conserved across the 3 different species. The organization is slightly different. What was really surprising, though, is when we were doing PCA analysis on global somatic cell populations from the 3 different species, we find that, as expected, for example, immune cell populations are clustered and highly correlated across the 3 different species, as well as endothelial cells. But when we are actually comparing somatic cells, the testis-specific somatic cells present within the testes, here we see a lot of variations in the transcriptome of these cells, and actually we see shifting in relationship when comparing, for example, smooth muscle cells in the testes that line the basement membrane are more highly related in monkey, but these cells actually tend to cluster with a different cell type when comparing them to mouse and macaque, to human and macaque. So what we see is that when comparing the somatic compartment, there's a lot of variation within signaling, and also sometimes the same exact cell types don't tend to cluster together. Instead, they cluster with different cell types within the testes. And so we repeated this analysis using ligand receptor analysis, and we find that there's similar, these variations are driven by ligand receptor changes in ligand receptor expression patterns that are observed in these different somatic cell types. FGF2 signaling is known to be important for spermatogonial stem cell population, for example, in all 3 species. However, we know that in the different organisms is that this ligand is actually provided from different cell types within the testes. So suggesting that there's variation in both the ligand and the receptor expression patterns and dynamics across spermatogenesis across the different species. And just to briefly summarize, we have identified equivalent states in spermatogonia, and we've linked morphologically and histologically equivalent populations and provided a molecular signature. Although the spermatogenesis process is conserved, there's a lot of variation or heterochrony in the process of spermatogenesis, and by creating a universal pseudotime, we could compare across germ cell populations. And we've also identified there's significant changes in the germline soma communication. And so some of our future work will focus on resolving or creating a spatially resolved transcriptome. We've identified molecular cell types, but we have to map these in space in order to understand what are the signals coming from the somatic cells and how is this going to affect the differentiation trajectory of germ cells. To do this, we're using intrinsic fish, which I'm not going to have eventually a lot of time. We've piloted this approach and began analyzing 80 meiotic genes across the in the seminiferous tubule of the testes, but these are very preliminary analysis and will be expanded to a larger panel of genes. And with that, I'd actually like to thank the people who have actually done the work. A lot of the single cell RNA-seq work was really led by several talented students, Adrian Shami in my lab, and also Shan Ying and Jun Lee's lab. This is a collaboration, a very close collaboration with Jun Lee, as well as the Orvig lab for providing primate tissue. The spatial work will be conducted by Gabe Mansky in my lab, Shan Ying, Morgan, and also a very close collaboration, of course, with Jun and Arvin Rao. And I'll take any questions.
Well, thank you, Sue. Great talk. We got a few questions. We're only going to have time for one, and the rest you will have to answer to each person. I'll just pick one here. Do your single cell RNA-seq support the out-of-testis hypothesis, which proposes that all the new genes are first expressed in testes?
Yeah. I think that's a really interesting question. So I think definitely in the testes, there's a larger number of transcripts that are detected, both coding and non-coding, that are present in the testes, but whether and they're not really observed in any other tissues. So there is a large gene diversity. I can't say we can suggest that maybe this could be a plausible idea, but we don't have any direct evidence for that.
Okay, well, thank you so much, Sue. And please answer those questions offline. Our next speaker is Justin Colacino. He is an assistant professor in the Department of Environmental Health Sciences in the School of Public Health. Justin is an expert on environmental exposures on stem cells. He's interested in the epigenetic reprogramming of stem cells in breast cancer development. Justin got his Bachelor of Science from the University of Texas in Austin. He did a master's program in public health at the University of Texas in Dallas. Then he came to Michigan. He did his PhD in the Department of Environmental Health Sciences, where he is now an assistant professor. And Justin will talk about single-cell transcriptomic profiling to understand breast stem cell heterogeneity in development and cancer disparities. Welcome, Justin.
Thanks, Matt. So thanks so much for the invitation to come present our work. Yeah, so I'm going to follow in Sue's footsteps here and continue the discussion of single-cell RNA sequencing data. Here, we're looking at the normal mammary gland as our target, and we're really trying to think about how do we take these data and then use them for translation, really to understand the biology of cancer disparities and also start to inform clinical approaches in cancer prevention. And so really where we're coming from is that this concept that cancer in general looks a lot like development gone awry, right? And so this top panel here, this is a histological depiction of just normal mammary gland development. Right after birth, the mammary gland consists of these really rudimentary terminal ducts. They're loaded with these sort of like undifferentiated stem-like cells. And then once the signals from puberty start to kick in, these cells will start to invade and grow and divide in the extracellular matrix of the breast, eventually making these alveolar ducts, and then making this really nice forking tree, like this lobule type 1 forking tree that you see at the end of puberty, right? And so we also see parallels with how cancers develop. So in cancer, we go from normal tissue, we also see this sort of like expansion in situ and this invasion. And these parallels between development and cancer have been noted for over a century, right? But only more recently, last 20 years or so, have we been able to do the molecular profiling necessary to really compare the transcriptomes and the protein profiles of stem cells, both in normal development and in cancer. And so what my lab's really interested in is understanding stem cell heterogeneity. And so I was working in Max Worsh's lab as a graduate student, and we would be getting reduction mammoplasty tissues from women getting voluntary breast reductions at the U of M hospital. And I would take these back to the lab and I would dissociate them to a single cell suspension, and then we would plate them in culture conditions that are conducive for stem cells to grow, these so-called mammosphere conditions. And you can count the number of mammospheres that form, and it's really a surrogate readout of the number of stem cells that are in that tissue. And what we were seeing in normal women is this massive variance in the number of stem cells from individual to individual. And maybe some of you have seen these controversial Vogelstein papers that have been in Science where they're talking about the number of stem cells and stem cell proliferation as a major driver of risk. But here you see these differences between individuals, and it really makes us wonder, why are there such big differences? And so our lab is really interested in trying to understand this heterogeneity in stem cells and how does this drive cancer risk. So this cartoon here just models, mammary gland development starting from an individual sort of green stem cell there. Maybe the mammary gland starts to develop, and maybe, at the end of development, we have a fully formed mammary gland with just a small proportion of stem cells in there. But if a stem cell starts to differentiate and divide in the presence of either genetic risk factors or environmental risk factors, this can alter stem cell differentiation trajectories, leading to a greater proportion of stem cells in the developed mammary gland. And so we know that this is the case, for instance, for women who inherit a BRCA mutation. They tend to have more normal-like stem cells in their mammary gland. We also know from toxicology studies that you can also induce more stem cells in the mammary gland via in utero exposures to endocrine active compounds like BPA. So estrogen mimicking compounds, these exposures in utero, when there's all of this development, can promote an expansion of the stem cell state. We think that this expansion of the stem cell state is a really important early stage in carcinogenesis. And so we really wanted to start to answer these questions, like how can we use these single cell methods like Sue was talking about to dissect stemness in the normal human mammary gland. And so the mammary gland is a relatively simple organ, just to orient you all here if you're not super familiar. But there's two major epithelial cell types in the mammary gland. There's myoepithelial cells and there's luminal cells. The luminal cells and the alveolar structures, their job is eventually to synthesize breast milk. Myoepithelial cells, they constrict. And then there's also subpopulations of sort of like stem cells that are there to support the growth of the mammary gland during pregnancy and puberty. And also to respond in case of injury. And so we were really interested in understanding the biology of these stem-like cells. And so this was back in 2013, the very early days of single cell biology. We took this project on it. And so we were collecting these dissociated reduction mammoplasty tissues. We break them down to a single cell suspension, doing a similar approach to what Sue was talking about, where we would FACS purify our stem cell population here expressing aldehyde dehydrogenases. But we started out using a different technology. We used this Fluidigm instrument, which would capture each individual cell into an individual little micro well. And then we were doing 96 gene qPCR for a specific set of stem cell targets, which was pretty exciting at the time, right? So this was back 2013, 2014. This was pretty hot. And this gave what seemed like a lot of data at the time, right? So we had 96 genes. We ended up profiling, somewhere around 100 cells, right? This was the early days, but we could look at the expression heterogeneity of these individual stem cells. Remember, all these were FACS purified. And we really saw what appeared to be 4 distinct clusters pop out. And this was all based on those 96 gene expression profiles. And so this was sort of curious to us, like, why do we see so much heterogeneity in stem cells in these populations? And so we could look at individual markers in a similar approach to what Sue was describing. And we could see that we have our cluster 3 here. These are like our more mesenchymal-like cells. They're expressing high levels of what we know are mesenchymal markers in the breast. Things like CD44, things like ZEB2, slug transcription factor, vimentin, twist, these like very like mesenchymal markers. We also saw that there was a subset of cells that had a very epithelial-like phenotype. So these had high levels of E-cadherin and MUC1 and the luminal transcription factor GATA3. But what we were most interested in was this cluster of these cells that appeared to be in this like hybrid state, right? They were expressing high levels of mesenchymal markers and high levels of epithelial markers at the same time, as well as high levels of some of our favorite stem cell markers like ALDH1A3 and CD133. So this really made us think, like, why is there so much heterogeneity? And is there any like biological relevance to cancer that we can pick up in this population, especially of these rare hybrid cells? And so we went and we dug into some of the publicly available TCGA data, and indeed, genes that are overexpressed in this hybrid epithelial mesenchymal cell population, they tend to be more expressed in triple negative breast cancer. So triple negative breast cancers, very aggressive subtype of breast cancer with no targeted therapy available. And then even within triple negative breast cancers, women that have higher expression of these hybrid EM marker genes have worse overall survival. And so what we're seeing here is that there might be some association between this sort of like primitive hybrid epithelial stem cell state and aggressive breast cancers, lending even more credence to this relationship between stemness and cancer, risking aggressive cancers. And so our next question was really trying to understand, how does stemness and these stemness states, how do they change throughout development? And then how does that mirror what's happening in cancer? And so to do this, we undertook another single cell experiment. And so here we are still working with those normal reduction mammoplasty tissues. We took those back to the lab, we dissociated them to single cells. And then some of those single cells we profiled via that Drop-seq method that Sue was talking about, that single cell RNA-sequencing method. Some of these we pushed into a more stem-like phenotype via the conditional reprogramming process. This is a technique for primary tissue culture that was developed by the Schlegel lab at Georgetown, where it really does a nice job of pushing your cells back into a more primitive state, to sort of like study these stemness alterations in vitro. And then we could compare and contrast, you know, like what do we see at baseline in these women versus what do we see after we've sort of like pushed these guys to a more stem-like state. And then we wanted to do an integrated analysis similar to what Sue was talking about, where we take advantage of this like rich set of publicly available data that describes mouse mammary gland development in utero, and then early in adulthood, and then in full adulthood, and then also what's happening during pregnancy. And so this was all work that was done by Tasha Tong, who's a great toxicology PhD student in my lab, Mike Brooks, who's a postdoc in Max Wish's lab, and then Yutong Wang, who is a electrical engineering and computer science PhD student who's just like a whiz and came up with all of these cool data set alignment tools, including this one, which we used in this paper called Corgi. And so we're really interested in trying to sort of like distill this really complicated single cell data down. And one of the ways that we're trying to do this is to you know, calculate the stemness of each individual cell. And so we came up with a relatively simple scoring mechanism here, where we can take all of the reads in an individual cell and then tally up how many of those reads are attributable to genes that we know are associated with an embryonic stem cell state. And so then we can compare and contrast, you know, the distribution of this stem cell score between different conditions. And so on this plot, we have NM, which means those normal mammary cells. Those are straight from the patient. They have a relatively low stem cell score, but then when we put them in those conditional reprogramming, like de-differentiation conditions, it cranks up our stem cell score. So this is like good proof of principle as we're trying to like optimize our techniques. We can also validate that that conditional reprogramming process causes an overexpression of some known mammary stem cell genes, sort of like doing some good gut checks about our single cell data, making sure everything's working as we would expect. But we really wanted to develop a new way to put our human results in the context of a pseudotime analysis, kind of like what Sue was talking about too. So we worked out this method, largely Yutong worked out this method, where we plot the developmental data. So from this Girardi et al. Sort of like developmental mammary gland atlas, where they have mouse mammary gland data on embryonic day 16, 18, postnatal day 0, 4, and then adulthood. And we can drop them on a PCA plot here, the top left, and they just sort of like fall out naturally in their own pseudotime from the most primitive in the top left, and then pseudotime kind of like moves down to the bottom right. And these also nicely branch into a luminal branch and a basal branch. And so this is like a cool way just like to see how these data just sort of like naturally fall out when you do these dimensional reduction techniques. We can then score each individual cell on its pseudotime and then calculate the pseudotime distribution. And like you would expect, as we're like marching forward in actual time, we actually get to march forward in pseudotime as well. So now we wanted to see, can we align our data on top of this mouse mammary transcriptome atlas and see like where do our data fall out. And it was quite interesting. We get to see, so this is the mouse data only, top left panel. Bottom panel is when we start to align the conditional reprogram data on top. And you can see that in general, these data are more resembling sort of like late embryonic, early postnatal expression patterns. But then we can compare and contrast with what we have for the normal mammary glands. So those are like the cells straight out of the patient. You can see that the normal mammary gland cells align quite nicely with adult mouse cells. And so we can calculate and compare and contrast the pseudotime of each of these. So we can, again, validate that our conditional reprogramming process is inducing a more differentially, or a more like developmentally immature phenotype. We're also curious about seeing how do these like stem cell phenotypes change throughout key developmental life stages in the mouse. And so this is a nice data set from Bach et al. Where they had done single cell RNA profiling of the mammary gland from nulliparous mice, meaning mice that had never had a child, mice that were mid-gestation, so they were currently pregnant, mice who were lactating, and then mice who had stopped lactating and undergone this natural process called post-involution. And so we can map those like mouse pregnancy data on top of the mouse developmental trajectory. And it's pretty interesting, I think. So the nulliparous mouse looks a lot, it has like an older pseudotime, looks more like an adult mouse. And then once the mouse is in gestation, pseudotime drops, right? It is a much more embryonic-like phenotype during gestation. You can see at lactation, it's getting older, but it still hasn't achieved like that adult-like phenotype until that post-involution process. And so we can also come back to that hybrid epithelial mesenchymal cell story, right, where we had previously seen in our fluidigm data that there was this rare subtype of hybrid cells that had this like epithelial mesenchymal phenotype at the same time. So here we scored cells based on their co-expression of EpCAM, an epithelial marker, and Bimentin, the mesenchymal marker. And then we could see that in the nulliparous animal, it's about 10% of cells meet that criteria. Those animals during gestation, that number jumps to almost a quarter of the cells. So we're seeing this like expansion of these hybrid cells. As we move through lactation, that number drops precipitously, and then post-involution, it's even lower. So this actually aligns quite nicely with our understanding of breast cancer, where there's a very aggressive subtype of breast cancer called pregnancy-associated breast cancer that happens in that like early stages, like that few months right after after a woman gives birth. But after a woman has given birth, 5, 10, 15 years later, her breast cancer risk decreases. And I think we're picking up that signature with respect to the stemness and differentiation state of the mammary gland. Taking what we knew about the stemness signature that we're picking up across the development, we wanted to then interrogate, how do human cancers look if we start to align these on top of the align these cancers on top of that same mouse developmental axis. And that's what we did here. It's over 1,000. This is bulk RNA sequencing of breast tumors from TCGA. So we can now like take bulk data and align it to the single cell trajectory, which is pretty cool. And you can see that in general, cancers tend to be more developmentally immature, right? None of them are really aligning with adult mammary glands, or like, which matches up with what we understand about cancer in general. But then basal cancers, these like triple negative, really aggressive cancers, they're the ones that are the most developmentally immature. And we can look at gene signatures of which genes are most inversely correlated with pseudotime. So the genes that are expressed highest in the sort of like top left component, these really developmentally immature cells. And we can see that a lot of these genes have a positive association with poor outcomes in breast cancer patients. So this gives us another idea that we're dealing with some really aggressive breast cancers that have reacquired stemness. And so next we wanted to see, can we start to translate these findings into understanding the biology of cancer disparities? So some of you may know that women of African ancestry in the US are much more likely to be diagnosed with the most aggressive subtype of breast cancer, that triple negative breast cancer, 2 to 3 times more likely. If an African American woman is diagnosed with TNBC, she's also more likely to die from the disease. And so these are like very profound breast cancer disparities that it's an extremely important public health problem. And so to get at this, we've been working with our colleagues at the Susan G. Komen Normal Tissue Bank, who will recruit healthy women to come and voluntarily give breast punch biopsies. And then those punch biopsies are available for experimental interrogation. So we've been collecting these punch biopsies from the Komen Tissue Bank. Down here is a picture of me and my graduate student, Shanice, when we worked at the tissue bank collection in Detroit. But we've been collecting these, and then we've been growing them up in culture for an experimental platform for assessment. And so this works okay. About a third of the tissues we can get them established in culture, but now we have a way to like test the effects of like diet or environment on stem cell phenotypes in a diverse range of normal breast epithelial cells. And so one of the advantages of this biobank, right, is that we have genetics, ancestry data on everybody, and we can confirm individuals' self-reported ancestry versus their genetic ancestry, sort of like a nice double check that people that are reporting as African American are of African ancestry, and people that are reporting as European American or European ancestry. And so we did single cell RNA-seq, working with the Wisha lab on 16 different samples. And so this is a T-SNE plot breaking this out. You can see that there's like some separation by race here, but really where we're seeing separation is based on whether a cell is a myoepithelial cell and expressing keratin 14 down here, or if it's a luminal cell and expressing keratin 8. So we're able to like pick up the two major cell populations in the breast after these processes. And then we can look at differential expression within these different cell fractions, right? So instead of having to deal with bulk breast tissue, now we can look specifically in luminal cells and be like, are there differences in the luminal cell gene expression signature in women of African ancestry versus European ancestry? And so we do see some differences. I'll just highlight a couple. So one is this gene CRYBB2, which has previously been associated with African American ancestry and ethnicity and survival in multiple cancer types. This is about 15 times upregulated in African American cells. It's also correlated with poorer outcomes in cancer regardless of race. Tends to be higher expressed in basal and HER2 breast cancers. And then some functional experiments in a nice paper in breast cancer research last year showed that overexpression enhances proliferation and tumorigenicity in triple negative breast cancer cell lines and xenografts, potentially pointing to like a functional gene that might in part explain why breast cancers in African American women are so much more aggressive. We can do a similar analysis in the myoepithelial cell fraction, those keratin 14 expressing cells. And so here we saw an interesting long non-coding RNA pop up, ESRG, this embryonic stem cell related long non-coding RNA. We know it's an early marker of induced pluripotency and really important for induced pluripotent programming. So something that we're interested in doing some functional experiments to try to dig in on. We can also go back to that stem cell score where we sort of like rank each cell based on its so-called stemness. And what we see is that for both the luminal and the myoepithelial cell fractions, the cells from African American women tended to be more stem cell-like. If you remember back to how we showed that basal breast cancers tend to be more stem cell-like, there might be something going on here where there might be some underlying differences in stem cell biology that are predisposing women of particularly West African ancestry to a more aggressive type of breast cancer. And so this is what we're really trying to figure out. And where we're moving forward here is thinking about, can we now start to target the stem cell state in women that have high proportions of stem cells in their breast, these like really high risk women, or other individuals like with other types of so like high risk cancers, familial cancers. Can we use methods and chemo preventives to target the stem cell state to reduce cancer risk? And so this is a study that we've been working on, a PhD student in my lab, Evan Hill, and research technician Anaga Tapaswi, where we're looking at the effects of a natural product from broccoli called sulforaphane. We've treated mice that are genetically predisposed to getting colon cancer with this, and then we've been doing some single cell profiling of their guts to really understand what are the impacts of sulforaphane on different cell populations to see, can we target the stem cell state? And what's really nice about these data is we can now look on a cell type specific basis to understand how are these chemopreventive compounds acting. We can also look at a more holistic level, like, hey, across all of the epithelium, do we see differences in the so-called stemness state? We can pair this with some patient-derived organoid work where we take patients with genetic risk, genetically inherited susceptibility to colon cancer, for instance, FAP, familial adenomatous polyposis patients. We take adenomas, and then we can grow those up in organoid cultures, and then start to dig out how do an individual cells respond, and then we can compare and contrast between the in vivo model and the in vitro model, ideally trying to translate this into a clinical trial. And so here we can come back to our higher level diagram. We can think about how are genetic factors, how are environmental factors perturbing the stem cell state, and then can we come in later in life at those high risk individuals, come in with a chemopreventive to try to bring that risk down, induce differentiation. And so there's a lot of people that work on this project. I'd like to thank all the folks in my lab, particularly Tasha and Evan, and Shanice for working on this, and Anaga, and then our funding sources, some of our collaborators in our single cell center have been amazing, and then some folks in the Wisha lab who have been also really essential to getting this work done. And with that, I would be happy to take any questions. Thank you.
Okay, thank you so much, Justin, for a great talk. I don't see any questions yet. I just may add a question myself then. So when you look at these gene expression changes in stem cells and between these developmental axes or between ethnic groups, obviously you're looking at the steady state RNA levels. Do you know, or would you predict that they are mostly driven by transcriptional changes, and how much would differential turnover of messenger RNA result in these gene expression changes that you see?
Oh yeah, I think that's an awesome question. No, that's not something that we have explored. And so we are doing some experiments. We're starting to perturb these cells in culture and seeing how they respond with respect to the stem cell state. But I know that your lab has developed some really interesting techniques to be able to model and measure those newly transcribed RNAs. I think that would be awesome. No, I have no idea. That's a great question.
All right. I don't see any other questions. So with that, then I would like to thank Sue. I'd like to thank Justin, and I'd like to thank everybody that listened in today. There will be no RNA Collaborative seminar 2 weeks from now, but in 4 weeks, there will be from the RNA Institute in Albany. So please tune in on October 7th when we will go to Albany for our next seminar. Thank you, everyone.